Good day, I'm Dr. T. Welcome to my office. Today, doing a Q&A video. Questions taken from class discussion boards and YouTube comments. Actually, all the questions come from my survey to class discussion boards. Okay, so first question, how do cells get the right sequence of nucleotides when replacing, or replicating, I should say, DNA? That's why there's the base pairing, so the ATGC. By having the hydrogen bonding, that gives you the right angle, the right width by doing a pyrimidine-purine pairing, thus the polymerase can correct it. So the way they get that is by the base pairs. Okay, where do new DNA nucleotides attach to the previous nucleotide in the strand, and does this create any problems for the cell? Okay, so yes, the new nucleotide is going to take its 5' end and attach it to the 3' end of the previous nucleotide. Now the problem, of course, is during that replication fork, one direction is going 3', 5', the other is going 5', 3'. So one side's going the way it wants to and can just chunk on no problem. The other one has to do a back stitch, which is where we get the Osaki fragments from. Okay, why would an organism wish to have two different versions of the same enzyme? So these are called isozymes. And you're going to want isozymes, a couple reasons. One, and kind of the major reason, would be control. So you might have two versions. One does not respond to a certain regulatory molecule where the other does. So we could imagine something where you've got an adrenaline and the regulatory cascade that creates from that. If you have, say, gluconeogenesis and glycolysis enzymes, you're going to want to respond differently in the liver versus the muscle. And then you could also have things like, a little bit different affinities. So we can look at hemoglobin, though once again, not technically an enzyme. You've got fetal hemoglobin, which is a little bit grabbier to the oxygen so that you can pull it from the mother hemoglobin. Okay, dominant recessive alleles don't have all the same thing taking place at the chemical level, making the dominant or recessive. I think I misread something. Anyways, however, at the chemical level, what things could be taking place leading one allele to be dominant over another recessive? So typically speaking, the recessive gene is not going to be giving you the functioning enzyme. So a dominant gene typically, but not always, is going to be giving you an enzyme that does something. So I think the example I did in one video was my eyes. I know that I am heterozygous for brown eyes. So I've got two genes, one from my mother, which creates an enzyme that creates a pigment in my eyes, and thus the irises are brown. I also have a gene from my father, which does not create an enzyme that makes a pigment that makes my eyes brown. Now, if I didn't have my mother's gene, then my eyes would be blue, because that's kind of the pigment-free color of an iris. But since I do have her gene, therefore I do produce pigment in my iris, and it is brown. Thus, brown eyes is dominant, blue eyes is recessive, because blue eye is really the lack of a functioning enzyme in that particular set. So you could either have a non-functioning enzyme either because the enzyme that has been mutated in such a way that there's a protein produced that just doesn't get the job done. Alternatively, you could have mutations in the regulatory regions that just lead to not having a protein to do the job. So yeah. So with that said, those are the questions for today, and I will see you in future videos. Have a good day.